Good day everyone. I am Rinald Y. Cabardo, a second-year student of Bachelor in Science of Medical Technology, Section B. In this video, we are going to talk about chromosome and its parts. Now, what is chromosome? And where can we find it? In order to answer these questions, we must delve into the very depths of our body, into our very own cell. Here we have a picture of a cell. By definition, cell is the building blocks of life. In a cell, there is a small region which is called the nucleus. In the nucleus, we have a very thin structure or thread-like structure which is called the chromatid. The chromatin carries or consists of DNA that carries the genetic material and protein, also called histone. Okay, in a chromatin, if we are going to zoom in the chromatin, we can find the very DNA of our body. Okay, this is the scientific illustration of a DNA. DNA is a double helix, consists of nucleotides. So, if we're going to ask, where is the chromosomes? Chromosomes will be visible only during mitosis. When we see mitosis, it is the division of a single cell into two identical daughter cells. Before entering into mitosis, the cell must enter first into a process called the interphase. The interphase, it is the phase between cell division. As you can see here, it is the cell life cycle wherein the cell must go first into the process of interphase before going to the process of mitosis. Mitosis is divided into prophase, metaphase, anaphase, and telophase. During interphase, the cell prepares to divide. It is also the phase where DNA replicates and centrioles duplicate. After the cell prepares for division, the chromatin condenses, coiled together, and became a chromosome, like this one. This is our chromatin. It's a thread-like structure. It's like a thread. And during mitosis, it condenses to become chromosome. So during mitosis, the chromatin condenses to form a chromatid. The chromatids are joined at the centromeres, this one, it is the centromeres, to form a single mitotic chromosomes. Now, we understand that chromosomes became visible during mitosis. So now we are going to determine the different parts of a chromosomes. A chromosomes is composed of two chromatids. This one. Chromatid are the coiled, dense form of chromatins. So if the two chromatid attach to each other, it is through the process where centromeres attach the two chromatids. Okay, when we see chromatid, it is the half of chromosomes, the densely coiled compact form of chromatid. In a chromosomes, we have centromeres. Okay, centromere, it is the region in chromosome where chromatids are linked. So there are different types of chromosomes. First, we have metacentric. Second, submetacentric. Third, acrocentric. And fourth, telocentric. So what are the difference between these types of chromosomes? When we say metacentric, the centromere is in the center of the chromosomes. As you can see here, the centromere is equally divided the chromosomes. When we say a chromosome has a part which is called the telomere. The telomere, it is the non-coding DNA found at the both ends of chromosomes. Its job is to protect the chromosomes from any damage of the genome. The other type of chromosomes is what we call the submetacentric. When we see submetacentric, the centromere is located near the middle. Okay, as you can see, the centromere is near in the middle. It divides the chromosome into an equal part. The upper part is shorter than the lower part. The short arm or the short part of the chromosome is called the P arm, and the longer part of the chromosome is called the Q arm. The next type of chromosome is called the acrocentric. When we say acrocentric, the centromere is in the near end of the chromosome. As you can see here, the centromere, it is in the near end of the chromosome. In a acrocentric, there is a part that we call satellite stalks. This one, it serves as identifying markers for other chromosomes. The last type of chromosomes is what we call the telocentric. When we say telocentric, the centromere is located at the very end of the chromosomes. So, in summarization, the different parts of chromosomes are chromatid, centromere, telomere, P arm and Q arm, and the satellite stalks. Since we're done differentiating the different parts of chromosomes, let us discuss mitosis. So, what is mitosis? Mitosis, it is a process of a cell division where a single cell divides into two identical daughter cells. Here we have an illustration of how mitosis happens. A single cell divides into two identical daughter cells. Its purpose is for growth or tissue repair. To understand fully what is mitosis, we need to understand first the cell life cycle. When we see cell life cycle, it is the changes of a cell undergoes from the time it is formed until it divides to produce two new cells. Here we have a diagram of cell life cycle. Cell life cycle is divided into two major events. First, the interphase, then the mitosis. In interphase, it is divided into three phases. So to understand this, we have to unlock what is interphase and what are the different phases in interphase. So interphase, it is the phase between cell division. In this phase, the cells prepares to divide, DNA is replicated, centrioles also duplicated. It is divided into three phases. We have the G1, which means first gap phases, the S, which means synthesis phases, G2, which means second gap phase. During G1 or first gap phases, the cell carries out routine metabolic activities. For example, making proteins. During synthesis phase, the DNA is replicated. And during G2 or second gap phase, the cell grows and it is now determined if can now proceed to mitosis. So after interphase, if the cell is fully developed and it is determined, now it can proceed to the process of mitosis or cell division. Mitosis is divided into 4 phases: the prophase, the metaphase, the anaphase, and the telophase. In prophase, this is what happens. The chromatin condenses. So as all we know that chromatin, it is a thread-like structures in the nucleus. But in mitosis, it condenses, it coils to form into chromosomes. Now after that, the centrioles, this one, divide and migrate to each pole of the cell, like this one. Then the microtubules, IA, that is to say, spindle fibers extends. So these are the extended spindle fibers of the centrioles, which is traveling in opposite direction towards the opposite poles of the cell. Nucleolus and nuclear envelope disappear. So this one, the nuclear envelope of nucleus will disappear in the late stage of prophase. After prophase, we have the phase of metaphase. So what happened during metaphase? During metaphase, the chromosomes align near the center of the cell. The nucleus is gone. The centrioles reach the different or the opposite poles of the cells, and the chromosomes are starting to align in the center of the cells. Then the spindle fibers are attached to the kinetochores of the chromosomes. So as we all know, chromosomes have centromeres, and centromeres have the kinetochores, that is the location where the spindle fibers are attached. After metaphase, we have the anaphase. During anaphase, the chromatids separates. So each chromosomes is composed of 2 chromatids. So during anaphase, those chromosomes were separated. So one sets of chromatids for the other side of the pole, and the other sets of chromatids to the other side of the pole. Now, the sets of chromosomes is moved by the spindle fibers towards the centrioles. So the one pulling the chromosomes were the spindle fibers from the centrioles. Each set of chromosomes reached the pole and cytoplasms began to divide. So during anaphase, these chromatids will reach the different poles of the cell, and the cytoplasm of the cell began to divide. This is where cytokinesis appear. Cytokinesis, it is the division of cells of cytoplasm to produce 2 new cells. It begins in anaphase and continues through telophase. So this, a little bit what we call cleavage furrow. This one, cleavage, it is the sign of cytokinesis, where the process of dividing the cytoplasm is starting. Cleavage furrow, this one, it is an identification of plasma membrane, and it is the first sign of cytokinesis. After anaphase, we have the telophase. In telophase, the nuclear envelope form around each set of chromosomes to form 2 separate nuclei. So, during prophase, the nuclear envelope of nucleus disappear. But during telophase, the nuclear envelope began to appear to divide the two nuclei. So, after that, after the nuclear envelope formed, the chromosomes began to uncoil. So, this is what happened if the chromosomes will begin to uncoil and the two new cells separated. So, in this picture, mitosis is complete, two different daughter cells is produced, and the new interphase began. So these are the phases or the process of mitosis that happens to our cells. Thank you.